you for the introduction, and thank you to the organizers for the invitation. I am very happy to be able to present part of my PhD work. So I am a third-year PhD student under the supervision of Amandine Véber for the mathematical part, and of Nathalie Machon, who is an ecologist at the National Museum of Natural History, so at the Jardin des Plantes, for the applications to ecology. Today, what I will present you is not the applications to ecology. I will focus on the more theoretical part of my work, which is motivated by the following experiment. So what you can see here is the result of an experiment by Hallatschek Nelson with the fluorescent bacteria and yeast, which have been put to grow into Petri dishes. So that's actually very close to the experiment Denafusco presented at the beginning of her talk on Monday. So you have bacteria which are fluorescent, and this fluorescent does not influence how the bacteria behave. It's just kind of a marker, and it's completely neutral. So the bacteria or yeast on the right are put in at the center of the Petri dish, and they are let to grow. And what you observe is that very quickly at the front, but still on a fairly large area actually, you observe a sectorization of the population. In some areas, all bacteria are green, and in others, all are red. Qualitatively, why these sectors appear is fairly well understood. That is because of stochasticity at the front edge, where you have very low population densities, which is simplified by the expansion. It's kind of serial founder effect, actually. And in one expansion direction, you lose one type, but in different directions, you potentially lose different types, and that's how you see the sectors appear. Yet, apart from this, quantitatively, questions such as how many sectors do we have, what is the width of the area in which we can see these sectors, and other more precise results. Actually, not much about that. That's partly due to the lack of stochastic population genetics models, continuous in space and adapted to expanding populations. This may sound a bit provocative at first, especially given the number of talks we got since the beginning of the week on expansions. But actually, if you think about it, among all these models, you think of the model which were stochastic, keeping the space continuous and adapted to expanding populations and to the study of genetic diversity. Actually, there were not that many models. So if we look in the literature on methods to study genealogies of expanding populations, you can find several approaches which are adapted to different questions. So one of them will be to define in advance, what will be the population growth dynamics, or more generally the population fluctuations, so that you can go backward in time to study the genealogies. So this approach is interesting if you don't want to actually study the properties of the expansion itself, since you have already determined what will be these properties. So another one will be to use branching processes in a continuous phase. But the one I considered during my PhD is the use of ghost individuals. So what do I mean by these ghost individuals? So just a fancy name put on a concept originating from interacting particle systems theory, and in particular processes such as the contact process, in which empty sites can be modeled as occupied by particles with a specific type, which I will refer to as the ghost type. So this concept from interacting particle systems has been first applied to population genetics in one article by Hallatschek Nelson with a study mostly by simulations, and in a more theoretical paper by Durrett Fan. So in the model they consider, the population is divided in dims or patches which are along a line, so it's in one dimension. And each patch can contain a maximal number of individuals, the kind of carrying capacity. And if your DIM contains less than this maximal number of individuals, then you fill it with ghost individuals. The reproduction dynamics is then given by what can be considered in interacting particle system as a biased voter model, and in population genetics as a Moran model with selection. And on top of it, you can add genetic diversity by putting a specific mark on real individuals. So the key features of this model, which can be generalized to others actually, is that the ghost individuals can reproduce as well in the model, which seems a bit weird from a biological viewpoint, but actually that's just a way to model stochastic fluctuations in population sizes. A ghost individual reproducing will just be a local extinction or decrease of population density due to stochasticity. For reproduction, these ghost individuals are in competition with real individuals, and since we actually want to observe an expansion, we need to give us a very strong selective disadvantage to ghost individuals against real individuals. Otherwise, you will just have an extinction. So, during my PhD, I've been applying this concept of ghost individuals to one population genetics process, which is continuous in space and in time, and which is called the spatial Lambda-Fleming Viot process, which we refer to as SLFV because it's shorter. So the main characteristic of this process is that, contrary to many other population genetics models where you have a reproduction dynamics, which is kind of individual-centered, you explain how each individual reproduces, potentially given who is around. So here instead we have a reproduction dynamics, which is kind of reproduction event-centered. So we have an underlying Poisson point process, which is giving reproduction events which occur in specific areas whose characteristics are given by the Poisson point process. So in what I will present you today, these areas are always balls whose radius is random and whose distribution satisfies the following property, but you can consider other shapes. And for instance, I've been considering ellipses instead of balls. That doesn't really change much, actually. So we will also consider that we are looking at the population from very, very far away at first. I mean, in the original model at least, so that we have an infinite number of individuals in each location. In the specific version of the SLFV I have been working with, we also need to consider that all the individuals at the same location have the same type. And then to define our reproduction dynamics, we will consider that whenever a reproduction event occurs, we choose one parent uniformly at random inside the ball. The parent reproduces, its descendants fill the ball, and the other individuals in the area die. Nothing occurs outside the area. So to illustrate this, we will consider that initially the population is in the following state. We have 4 different types, and in each location, all individuals have the same type. So we wait until a first reproduction event occurs. So that's this one. The affected area is here, and the parent sample is red. So the ball is filled with red individuals. And we'll repeat that again and again. So next time, the area is this one, the parent is white, so we fill the ball with white individuals. Of course, as you may have already noticed, this construction is only informal, because here it works because my slides are compact, so the reproduction events occur at a finite rate, but the complete model is on RD, so you have an infinite number of reproduction events occurring at each instant. So if you want to define the process formally, you have several ways of doing it. I think the original one was to define it as the unique dual process associated what can be considered as a process encoding genealogies, which was done in a paper by Barton, and Véber. You can also define your process as the unique solution to a martingale problem, which is often the approach which is the easiest to use to prove results on the process after. And you have an alternative approach introduced in a paper by Véber and Kolbinger, which is close to the informal construction with a Poisson point process, but you extend it to also sample the parents at the same time. So I won't go more into the details of the formal construction, but I will still explain you fairly quickly what is actually the state space the process is defined. So in order to make notations readable, I will consider that we have only two types in the population, the blue type and the gray type. And we will consider the set of all measures on RD times our type space, such as we can define what can be considered as a density in gray individuals, and such as this density is 0, 1 valued. So, I mean the set, not the interval, such as we can consider that in each location, all individuals of the same type. So, of course, I said there exists a density and not a unique density, and that's where all the problems occur actually, is that this density of gray individuals is not uniquely defined for a given measure. So we can't assign uniquely a type to a given location. And so the complete state space is the Cadillac space on this set of measures equipped with standard topology we put on sets of Cadillac paths. So now that I have presented the original model, I'll explain how we can add ghost individuals in it. Because currently, I mean, we have a model with an infinite number of individuals in each location. So, I mean, the expansion already occurred. I mean, the space is already full. So we need to modify it somehow to be able to have expanding populations. So this time we will consider that we have occupied and empty areas. The occupied areas still contain an infinite number of individuals, but the empty areas are filled with an infinite number of ghost individuals this time. And in order to give a selective advantage to real individuals, we will consider that whenever reproduction occurs, we sample k potential parents, with k potentially very large. We fix it in advance, but that's it. And then we choose our actual parent out of these k potential parents. If all the potential parents are ghost individuals, then the actual parent is going to be ghost as well. But if at least one of the potential parents is real, then the actual parent is going to be the first real parent sampled. So in order to illustrate it, we'll start from the same initial condition as before, but this time the white areas are empty, so filled with ghost individuals. We wait again until the first reproduction event occurs, and this time we sample, I mean, I've taken k equal 3 this time, potential parents. Very well chosen example, so there is exactly one real potential parent, so it is the actual parent, it reproduces. The interest of the process lies in cases during which we have a reproduction event intersecting partly an occupied area and an empty area, but all the potential parent samples are ghosts, so we have what can be considered as a local extinction at the foot. And if several real potential parents are sampled, we choose one of them as the actual parent. So this time it's going to be the blue individual. So again, this is an informal construction, but you can totally generalize it. And actually, if you change the terminology and just use a standard terminology with like selection against ghost individuals, that's the standard SLFV with selection, but with new names put on it. So you can look at the corresponding references. So if you want to study genetic diversity in this kind of process, you can use the dual process, which encodes genealogy. So in case there are people here not familiar with what is a dual process, that's something which allows you to go backwards in time. So you can use it to show that the solution is unique, because if you can go backwards in time, that implies that you have only one way to go forwards. And in population genetics, it also encodes the genealogies in a sample of individuals. Indeed, if two individuals have a common ancestor, if there are no mutations, they have the same type, and if there are mutations, I mean, they have only occurred after this common ancestor. However, if we have ghost individuals, then it's a bit harder than usual to study genetic diversity, because we actually have two levels of genetic diversity with big brackets. One level, which is real individuals versus ghost individuals, which is, from a mathematical viewpoint, is studied just as genetic diversity, but actually that amounts to studying the gross properties of the subpopulation of real individuals. So to study the gross properties of the expansion. And then you have a second level, which is the genetic diversity among real individuals. And so the genetic diversity you actually observe. And in order to tackle this second question, you need to answer for the first one. So that's the one I've been focusing on during my PhD. So to illustrate how to construct this dual process, we can consider that we have several locations, and we want to know if at least one of them is actually occupied, so contains real individuals. So what we are going to do is we go backwards in time, and we look at the reproduction events which affected this sample. So for instance, the first one was this one, which affected exactly 2 individuals which come from the same parent. Now that we know that the 3 potential parents are these ones, but in order to decide which one of them is the actual parent, we need to know whether these potential parents are real or ghosts. But that's actually to know this kind of information that we started going backwards in time. So we don't have it. However, actually, we don't care because we don't need to know which one is the actual parent. We just need to know if at least one of them is real. That's totally equivalent to the descendants being real. So we add the 3 of them to our sample, and we continue going backwards in time. So these 3 individuals have the same parent, which is one of these 3 potential parents, etc., etc. So we can define our process formally. And we have the following duality relation, which is the same as, that's exactly the one for the SLFV section in the article by Henri, Véber, and Liu, in the case k equals 2, but you can very easily generalize it to k being larger. So this duality relation just tells you that a sample of individuals contain at least one real individual, if and only if at least one potential ancestor of the sample is real. And you need to have a more complicated equation with integrals because your type in a given location is not uniquely defined. So now for those of you who are already familiar with the SLFV, you must be kind of wondering actually what did I do from a mathematical viewpoint, apart from just putting a different terminology on an existing model. So actually, so the thing is, considering ghost individuals, which do not exist, actually motivates looking at a regime of strong selection or very strong selection against these ghost individuals, while the regimes which are usually considered in the literature are more like weak selection regimes. So I have been focusing on the regime in which your number of potential parents that you sample inside the reproduction balls grow to +infinity. So intuitively, what happens is that when you have a reproduction event intersecting only partly an area actually occupied, if your number of potential parents is finite, you always have a non-zero probability of having only ghost potential parents. But when your number of potential parents grows to +infinity, this no longer happens. And in the limit, if real individuals occupy the non-zero fraction of the ball, then one of them is bound to be chosen as a potential parent at some point. So in what we consider to be the limiting process, we no longer have extinction events due to stochasticity in reproduction. So that's for the intuition behind the limit. So my goal was like twofold. First, define what we have conjectured to be the limiting process with like the intuition, what happened, as a unique solution to a martingale problem, and also make the process actually appear as the limit of the k-parent SLFV when k goes to +infinity. So not just say that intuitively it seems to be the limit, really have a convergence result. So in order to do so, like use the standard proof for showing that a martingale problem admits a unique solution. So first, show that the martingale problem admits at least one solution. And this solution was constructed at the limit of a sequence of coupled k-parent SLFVs, which also fulfilled the second goal directly. And the second step was to construct what we wanted to be the dual process, and then establish the duality relation to show that our martingale problem had exactly one solution. And actually, the first step worked really well, and the second step actually failed in the more general case, because if we consider like the more general condition in the distribution of radiuses, which I initially introduced, and which ensures that a given location is affected by a reproduction event at a finite rate, then if I only have this condition, then the limiting dual process is not always defined. It's not enough to control the jump rate of the dual process. We need to put a structure condition to ensure the existence of the dual process. But under this stricter condition, I mean, the step 3 goes really well. So we end up with two different characterizations of the limiting process. So a first construction has the limit of a sequence of coupled k-parent SLFVs, which adapts an approach from the article of Véber and Wagelwinger, where we just extend the Poisson point process in order to sample at the same time as the reproduction event all the locations for the potential parents. So for the two-parent SLFV, we take the first two locations sampled for like the 42 parent SLFV, 42 first locations, etc. So they are all coupled together, and that ensures we can, if we decide initially one density, we can just have decreasing these densities, and we have a limit. And the limiting process is also a solution to the martingale problem, but potentially not the unique one. So to show that the martingale problem has a unique solution, so as I said, we use the dual process, which is actually not always well defined, and to control its jump rate, to obtain a kind of very technical condition which is related to the non-explosion rate, non-explosion of a branching process. But actually, this condition is satisfied if the radius of reproduction events is bounded, which is the case in which people usually work. So it's not that much of a problem, actually. And then, from a modeling viewpoint, one interest of the limiting process is that it seems to be a space continuous equivalent of another population growth model, stochastic population growth model, which is the Eden growth model, which is kind of well accepted as a stochastic population growth models for colonies of bacteria, for instance, but which, at least in its initial versions, is discrete in space and makes it hard to study genetic diversity. So here we have a process which looks like the Eden growth model, but in a space continuous version, and associated to tools which potentially make it easier to study genetic diversity. So with Amandine, we wanted to check if the process could actually be considered as an equivalent of the Eden growth model, and in particular, if it satisfied similar growth properties. So for this, we first had a look at what was known of the growth properties of the Eden growth model, and the answer is not that much, actually. So it grows at a linear speed, which is unknown. There are approximations with simulations, but no exact value. The fluctuations of the front positions are conjectured to be in the equivalence class of the KPZ equation, but that's only a conjecture. So there was not much to check. So in an article which will soon, in near future, be on arXiv, we showed that that's actually also the case for our limiting process. If we have an expansion starting from a half plane and we go along a line which is perpendicular to the half plane, we go to points further and further away. Then the expansion along this line is linear. And we here, for instance, to say when a point is reached by the expansion, we can't just look at the density because it's not uniquely defined. So we take balls smaller and smaller centered on this point, and we look at whether it contains real individuals or not to conclude. And so we have a linear speed as well. And just as in the Eden growth model, we don't have an exact value for the speed of growth. Okay, so what happens here is that the expansion is actually, initially the population is in the area that we do not see, so above the picture. And the area which is in purple is actually empty. It was supposed to be white or black, but I'm bad at coding, so it will be purple. So we have an expansion starting from here, and we simulate the expansion, and we see that it advances. And what we observe is that at the front edge, we have less and less different types, just as in the initial experiment. So initially, we had an infinite number of types to have something more visual. So we have like many different types here, and we lose them one after another in the front. Less and less of them. And that's it for the cool GIF not moving by itself. Thank you for your attention.
Thank you. It was very nice. I was wondering, in the last GIF that didn't move by itself, was there any, there's no selection against any types?
No, yeah. All the types are, the subtypes of real individuals are completely neutral.
Okay. But it could be, so you could extend, for now, you just have a big advantage of real individuals against ghosts, but you could potentially include small selection against
different types. Potentially. Currently, even the neutral case is complicated, but in the less near future, perhaps.
Okay, and about the relationship with the Eden process, you said that you don't know there's a speed, a constant speed, but you don't can't identify it
we have, for the speed, we have a lower bound. We were initially very happy about the lower bound, and then we did simulations, and the only positive thing is that it's the same order of magnitude. But the actual speed is almost 2.6 times faster than what we had with our lower bound, so it's pretty bad actually.
Okay. Is it easier and on fluctuation of the front, is it easier with your continuous your process than?
Currently we have 0 results on the fluctuations, so
no,
I can't really say if it's easier or not, but it's not easy.
Okay, thank you.
Any other questions?
Thank you. So I totally understand that you are interested in the 2D case for mimicking the experiment. What about the 1D case? Does it make sense? And if it makes sense, is it easier, for example, to get the speed expansion? And if so, do you have some intuition that the speed expansion should be the same in 1D and in 2D?
That's a very good question, actually. That's probably way easier to get the speed in 1D indeed, because in 2D what happens is that perhaps I can show on the illustration actually. So what you can see, in fact, is that sometimes in 2D you have some spikes which then grow sideways. So if you want to go in one direction, you can go straight, or you can kind of have a spike elsewhere which goes there. And that's really why it makes the study more complicated for the speed of growth, because you can go sideways and then back. And that will not occur in 1D. So, however, we have also been, to explain the discrepancy between our lower bound and the actual speed of growth, we have considered like a very simple toy model with Amandine, where we have two piles of cubes in which we have cubes falling, and the piles can also grow sideways. And we compared the speed of growth of just one pile or two interacting piles. And even with only two piles, the speed of growth of like one pile is significantly higher than without this interaction. So in this case here, when we can consider that we have many piles one after another, it's going to be even worse. So I don't think the speed of growth in 1D will be even close to the one in 2D, which is sad.